Ladies and gentlemen, it is my great pleasure and honor to declare the opening of today's symposium titled The Second Korea-Sweden R&D Network Day. First of all, I would like to thank all of you for your presence here today. I am Do-young Park, Director General of NRF. To begin today's symposium, I would like to introduce Professor Jung-hae, the President of NRF. She is going to deliver the welcoming remarks. President Ro, please.
Greetings, dear colleagues and friends. I'm Jung-hae, President of the National Research Foundation of Korea. I would like to extend my heartfelt welcome to all of the distinguished participants joining us today. This event marks the second Korea-Sweden R&D Network Day and follows the successful first one, which took place in Stockholm last year. The first Korea-Sweden R&D Network Day provided attendees from both countries a satisfying opportunity to share their research interests in person and expand their research networks and community. It is for this reason that we all are here today, and I hope we will continue to meet year after year. This year's conference has been virtually prepared due to COVID-19 pandemic, which has sharply changed our daily life and compelled us to adapt to the new normal. I thank Dr. Sven Stafström very much for his support and dedication to continue this networking conferences. I'm very honored to have this many eminent researchers and promising scientists from both countries here with us. Now I'm happy to introduce speakers from Korea. Professor Nari Kim from Seoul National University, who is also the director of the Center for RNA Research in the Institute for Basic Science, will give a keynote speech on her recent findings about the SARS-CoV-2 virus. Presenting in the session on COVID-19, Dr. Kim Seung-taek from the Institute Pasteur Korea, Professor Sung Beng-min from Yonsei University, and Professor Park Ne-ung from Archie University will give us many insights about how to cope with this pandemic. In presenting in the session on material science, Professor Lee Han from Korea University, Professor Hwang Byung-il from Chungang University, and Dr. Kim Seung-chul from the Korea Institute of Science and Technology will introduce us to the front line of novel materials. I'm confident that their contributions will serve as a sturdy bridge to stimulate international cooperation between our two countries in science and technology. Today's networking will facilitate exchanging insightful ideas and possible collaboration opportunities among scientists from both countries. And I hope it will serve as an example to demonstrate that international cooperation is one of the most powerful ways to tackle our common global concerns and to promote our common interests. Since establishing diplomatic relations in 1959, Korea and Sweden have sought to foster close ties through cooperation in diverse fields. Most recently, President Moon Jae-in made a state visit to Sweden last year to commemorate the 60-year anniversary of bilateral diplomacy between our two countries. With strengthening collaboration in areas such as future cars, biohealth, and 5G communications, leaders from our two nations share a consensus on promoting close cooperation in science and technology. Based on the strong partnership between the NRF and VR, we, the two funding agencies, will together be launching 4 new cooperative research projects on COVID-19 this coming December. I'm grateful that VR suggested the proposal, and I'm happy that NRF could respond in a timely manner to boost research collaboration between our two countries. I'm more than confident that the outcome of these projects will help us better cope with the pandemic. Today's conference provides us a valuable opportunity to bring researchers together to discuss innovative solutions and exchange views. By continuing to promote communications this way, we can be better prepared and make better future. Once again, I would like to express my appreciation to all the participants and especially to those of you who made every effort to make this virtual conference a success. Thank you very much.
President Ro. Next, let me introduce Professor Sven Staffelström, the Director General of BR. He will deliver the congratulatory remarks. Please, Professor Staffelström.
Thank you so much. Dear Dr. Rowe, dear participants, good morning from Sweden. It's still dawn here in Linköping, where I am for the moment. And as you know, we are in a difficult situation with the COVID-19 pandemic. We don't have a complete lockdown, but it's quite close. So most of us, we work from home. I think in this situation, it's very stimulating to have contacts with international colleagues. And it's my great pleasure to participate in this year's Digital Korea-Sweden R&D Network Day. But I must say that I would have enjoyed even more a physical meeting in Seoul, which we were originally planned for. But the way we work, and in particular the way we organize meetings, has really changed since the COVID-19 pandemic started. And we simply have to accept the fact that today it's not possible to travel the way we used to. The pandemic has in a dramatic way showed the importance of both fundamental understanding of biological systems and of research to create new knowledge. Thanks to years of research, we have a reservoir of knowledge about viruses and the way they spread and cause infections. But we had no detailed knowledge about the particular coronavirus and how to prevent or cure the patients from the COVID-19. The unprecedented efforts from physicians and from scientists all over the world have led to a situation which still is.
Men ni använder inte den. Ni har bokningen annat. Ja, men då så. Jag skrämde mig lite där, ja.
Someone has a microphone unmuted. Could you please check all your microphones? So unprecedented efforts from physicians and scientists all over the world have led to a situation which still is difficult in many parts of our world, but where we can see some light in the tunnel. I think the experience from the ongoing crisis regarding research is something that will affect research policy in the future. So what we need is really a resilient and sustainable research funding system, and also to a large extent a very flexible one. The COVID-19 pandemic has also affected the bilateral research program between Korea and Sweden. At a quite early stage of the pandemic, we made a common decision to focus this year's call on COVID-19. During a crisis, it is important to be flexible, and we, both NRF and VR, showed, I think, a very good example of this in the way we handle our collaboration. I said it when we met at the last network day, the first network day that was held in Stockholm, that Dr. Rowe mentioned, that was in June 2019. That coincided also with the celebration of 60 years of diplomatic relation between our countries. We find the collaboration with NRF extremely important. I stress it, extremely important, and also very well functioning, thanks to all the work that goes on both sides. And I think this dedication of focusing on international collaboration is so important, both in these days and in general as concerned research. I think we share a common view of the importance of international relations in order to build resilient and sustainable research systems. And this network day is another example of fruitful collaboration. I would like to sincerely thank Dr. Rowe and her staff at NRF for organizing this important event. And I'm really looking forward to hear, I won't be able to say during the whole day, but I'm really looking forward to hear some of the presentations. And I wish us all a very successful meeting. Thank you very much.
Thank you, Professor Staffelström. Next, I would like to introduce Professor Neri Kim from Seoul National University and Director of the Center for RNA Research at the Institute for Basic Science. Today, she is going to deliver a keynote speech titled RNA as a regulator, pathogen, and medicine. Professor Kim, please.
Thank you for the kind invitation. I'm Mary Kim at Seoul National University and RNA Research Center at IBS. It is my great pleasure to meet you and share with you some of the important progresses made in the field of molecular biology, specifically that involves a type of biomolecule called ribonucleic acid or RNA. I think this topic is particularly relevant today because SARS-CoV-2, the virus that causes the COVID-19 pandemic, is an RNA virus, meaning that the genome of SARS-CoV-2 is made of RNA rather than DNA. In fact, RNAs are highly versatile molecules, and they play key roles in various ways in all living organisms, from viruses to humans. Furthermore, in recent years, we have witnessed amazing technical breakthroughs based on RNA. For example, an RNA vaccine gave highly promising results in clinical trials for the COVID-19 vaccine, as reported last week by Pfizer. RNA is similar to DNA in its chemical composition and structure. Like DNA, it contains 4 different bases whose sequences encode genetic information. But unlike DNA, RNA is generated as a single-stranded molecule, and it can also form complex structures to gain various functions. It can also interact and regulate other molecules like nucleic acids, DNA or RNA, proteins or metabolites with high specificity. So RNAs can play a wide array of biological roles in cells. One classical example of RNA as a messenger is as a messenger of genetic information in protein synthesis. Genetic information is stored in DNA and then copied to RNA, which mediates protein synthesis using its genetic information. These so-called protein-coding RNAs are called messenger RNAs or mRNAs. There are many other types of RNAs that that have been also discovered. These so-called non-coding RNAs can either help protein synthesis or inhibit protein synthesis. Some of these RNAs constitutively function, while others are present only in certain conditions and cell types, playing regulatory functions. One important class of these regulatory RNAs is called microRNAs, which my laboratory has been working on for many years. These RNAs regulate virtually every cellular processes in development and pathology. One can even make an artificial microRNA to selectively control a gene of interest. This technique, called RNA interference, is a powerful tool to modulate cellular function and treat diseases. These artificial microRNAs are called small interfering RNAs or siRNAs. The first drug based on siRNA was approved by FDA 2 years ago. So RNAs are good, but not always good. One problem with RNA is that if you have a defect in RNA metabolism, that can often lead to genetic disorders. As many as 20% of human inherited disorders are due to RNA defects. So very active research efforts have been dedicated to develop ways to correct such errors in RNA processing. A new therapeutic approach using antisense oligos or antisense drugs that can target a select mRNA has been introduced for the treatment of genetic disorders like spinal muscular atrophy lately. This is a promising technology that is based on very deep knowledge of RNA biology. Another problem with RNA is something that we are experiencing right now. Coronavirus is essentially an RNA molecule wrapped in proteins and lipids. RNA replication is often error-prone, so RNA viruses generally undergo mutations more readily than DNA viruses do. This propensity for rapid mutation results in a frequent emergence of new RNA viruses, as well as a failure of pre-existing antiviral drugs. There are many RNA viruses that we need to pay special attention to, even after the current pandemic is over, because many RNA viruses will almost certainly re-emerge in the future. There are many mammalian viruses with RNA genome that include influenza A, HIV, Zika, Ebola, dengue, yellow fever, and MERS-CoV, and now SARS-CoV-2. To defend against these viruses, animal cells have evolved cellular defense mechanisms to directly recognize and eliminate the invading RNAs of extracellular origin by using proteins like toll-like receptors, RIG-I family proteins, PKR, OAS, etc., which are collectively known as innate immune system. Lately, in the face of pandemic, I have been working with my colleagues at IBS and Seoul National University, trying to understand how SARS-CoV-2 replicates and synthesizes its RNAs, and how the host defense mechanisms work against the virus. I would like to briefly highlight two of our recent studies. Firstly, my lab and Hyeshik Chang's lab at IBS RNA Research Center collaborated with KCDC to analyze the RNAs of SARS-CoV-2 using two complementary sequencing methods, namely nanopore direct RNA sequencing and DNA nanopore sequencing. From these analyses, we were able to map the transcriptome and genome of SARS-CoV-2. We defined 10 canonical abundant viral RNAs, including one genomic RNA plus 9 subgenomic RNAs that encode viral proteins from S through N. We also learned that the viral transcriptome is highly complex due to frequent recombination events, and found many non-canonical viral RNAs with unknown open reading frames, with deletions, truncations, and frame shifts, which would merit further investigation. We were also able to monitor RNA modifications on viral RNAs, including the RNA tail. These informations provide the framework for the molecular genetics of SARS-CoV-2. More recently, we have performed proteomic analysis on coronaviruses to identify the host vectors that interact directly with coronaviral RNAs. By RNA affinity purification using antisense oligos against viral RNAs and mass spectrometry, we identified 109 human proteins that bind directly to SARS-CoV-2 RNAs. Among these proteins, we found antiviral vectors that restrict viral replication, like LARP1, PARP12, ZEP, and FUBP3, etc. We also found proviral vectors that are required for efficient viral growth, like EIF3 proteins. We hope that this study will open new avenues for SARS-CoV-2 research and hopefully help the development of new antiviral therapy. As you can see, and as I explained to you, RNAs are not only integral parts of living organisms, but they can also make a pathogen that invade and cause devastating diseases. So RNAs are really powerful molecules. But can we actually use this power of RNA for the benefits of humanity? Before I conclude my talk, I would like to touch upon an important question of what we can expect from RNA research with respect to biomedical applications in coming years. Firstly, as you can imagine, RNAs are targeted for diagnosis, as in the case of viral tests. Viral RNAs are detected by qRT-PCR, which is the standard COVID-19 test to detect viral RNAs. A number of other new technologies like isothermal quantification are being developed to detect RNAs more rapidly. Plus, beyond viral diagnosis, molecular tests on RNA will certainly be expanded because RNAs give the most quantitative and sensitive means to monitor the state of cells and body. Less invasive sampling and less time-consuming and simpler detection will be necessary before RNA tests can be applied more generally. Secondly, RNAs and their derivatives can serve as therapeutics. Traditionally, most drugs are made of either small molecules or proteins like antibodies. But they take time, years, if not decades, to develop such drugs, and the cost of development is very high. So it is almost impossible to expect drug development for a rare disease with a small market size. RNA drugs, on the other hand, can overcome these problems. RNA drugs rely on the specific base pairing with the target molecule. So they are programmable, meaning that one can design the RNA drug based on the genetic sequences. So once you have the genetic sequences of a target molecule, one can use the same platform to design and manufacture various RNA drugs. So one can drastically reduce both duration and cost of development and production. There are currently 3 types of RNA drugs in the market or in clinical trials. I already mentioned siRNA, which is a type of artificial microRNA. siRNA can suppress a gene of interest to achieve a therapeutic effect. Antisense oligo is similar, but it usually can inhibit both inhibit and promote gene expression, usually in the nucleus by acting on mRNAs or pre-mRNAs. These 2 classes of drugs have already been approved and used for patients with genetic disorders. The third class, synthetic mRNA, is attracting a lot of attention these days, as this approach is potentially very useful for vaccine development. mRNA vaccine is basically an mRNA that can express a viral protein, which can act as an antigen to elicit immune response for the virus. As for COVID-19 vaccines, the viral spike protein is expressed as an antigen. mRNA vaccine has many advantages over conventional vaccines. Most of all, it takes just a few weeks for initial development, and manufacturing is much simpler because RNA is produced by in vitro transcription rather than from cells. And one can use the same manufacturing pipeline for multiple mRNAs, so it is scalable and flexible. Importantly, the mRNA vaccine is safe, likely because it stays in the body transiently. The efficacy, as reported by Moderna and Pfizer, seems very high, almost surprisingly high. So there is a high hope for this vaccine development. This mRNA technique can actually go well beyond vaccine. This is a new way of delivering genes into the human body. Compared to previous methods of gene therapy, like viral delivery or plasma DNA. This is clearly more promising for many reasons, including efficacy and safety. So I'm very excited about the possibility that mRNAs can make a major breakthrough in gene therapy. Of course, there are many technical limitations to overcome. This is only a very early days of this technology. RNA delivery should be improved in both efficacy and cell type specificity. Design of mRNA has also a very large room for improvement to increase gene expression and RNA stability. There are other types of potential RNA drugs. CRISPR is definitely one of them. So now we have a great toolbox for RNA drugs, and very likely this list will continue to grow to include yet unknown exciting new modules. I imagine that one can and continue to discover additional new types of RNA molecules that can be tamed to control gene expression. So the continued efforts to investigate the fundamental biology of RNA can offer new opportunities to develop next generation RNA drugs. With that, I would like to thank you for your attention and thank the people who contributed to our study. Thank you.
Thank you, Professor Neri Kim. Before we move to the session, we will have a 10-minute break. After the break, we are going to have two parallel sessions, COVID-19 and material science. Now, I would like to ask Korean speakers for COVID-19 to please remain this room, and the Korean speakers for material science, please move to conference room E3 beside this room. We also have an announcement for Swedish participants. For all Swedish speakers and attendees for COVID-19, please continue to stay same link. Whereas speakers and participants for material science, please access the other link that has already been provided to you. Finally, to keep the symposium proceeding efficiently, I would like to ask Professor Baek-Lin Sung to take the moderator role for COVID-19 and Professor Honey to take the moderator role for material science. Now, we are going to have a 10-minute break. Thank you for your attention during the opening session. Thank you.